Hello all. In this video, we'll look at the question number 25 of GATE Biotechnology 2015 paper. So the question is, which one of the following is incorrect about a typical apoptotic cell? Option A, phosphatidylserine is presented on the outer cell surface. Option B, cytochrome C is released from mitochondria. Option C, mitochondrial membrane potential does not change. And option D, annexin V binds to the cell surface. So to answer the given question, let's understand about the characteristics of apoptotic cells. So briefly, apoptosis is a process of programmed cell death that eliminates infected cells, cancerous cells, or aged cells. So these cells that undergo apoptosis are apoptotic cells. They undergo certain morphological and biochemical changes that are characteristic to these cells. So one of the characteristics is the structure of the membrane lipids. So XRP8 is a protein that acts as apoptosis-induced lipid scramblase. So scramblase is an enzyme present in the cell membrane that can transport, that is, scramble the negatively charged phospholipids from the inner leaflet to the outer leaflet and vice versa. So under normal conditions, the combined action of multiple mechanisms, including the activity of enzymes like flipases and flopases, maintains lipid asymmetry between the outer and inner leaflets of the plasma membrane. So flipases are enzymes that catalyze the transport of lipids towards the cytoplasm and require ATP, whereas flopases are enzymes that catalyze the ATP-dependent transport of lipids away from the cytoplasm, and scramblases are enzymes like XRP8 that catalyze the bidirectional non-energy dependent transport of lipids either way, both in and out of the membrane. So once the apoptotic program is activated, caspase 3 and 7 are able to cleave and activate the XRP8 protein, which acts as a lipid scramblase and leads to the loss of lipid asymmetry, resulting in phosphatidylserine exposure to the extracellular space. So phosphatidylserine are denoted by red circles here. So this rearrangement of the membrane lipids acts as an eat me signal that will allow phagocytosis of post-apoptotic cell corpses. So the apoptotic cells have phosphatidylserine in their outer surface. So if you look at this normal cell, there's no red circles on the outer surface. So this is one of the characteristic features of apoptotic cells. The next is cytochrome C release. So in healthy cells of vertebrates, the apoptotic protease activating factor 1, that is APAF1, is in an auto-inhibited form, and any basally processed caspase 9 and caspase 3 or 7 are bound by XIAP and hence they remain inactive. So upon apoptotic signaling, BH3 only proteins are either upregulated transcriptionally or activated through post-translational modifications. So the BH3 only proteins then bind to the anti-apoptotic BCL2 proteins to remove their inhibitory effect or activate back directly. So protein-lipid interaction might also be involved in the activation, which leads to their oligomerization and triggers the release of cytochrome C. It also releases SMAC-D, DIA-BLO, ENDO-G, AIF, and OMI-HTRA2. So all these are apoptotic factors that are released from the mitochondria. So the cytosolic cytochrome C that has been released from the mitochondria then binds to the APAF1 to induce the formation of apoptosomes, leading to caspase 9 and caspase 3 and 7 activation. These other proteins bind to XIAP to remove its inhibitory effect on caspase activations. So these activated caspases can then cleave XPR3 to rearrange membrane lipids that enables recognition by phagocytic cells, like we just saw in the previous slide. and in several other means to initiate the apoptotic cascade. So, cytochrome C release from the mitochondria is a crucial step in apoptosis. So, another feature is the mitochondrial membrane potential, abbreviated as MMP. So, during apoptosis, the mitochondrial membrane potential decreases. Although this is not entirely studied, there are several proposed models that explain the mechanism of the release of contents from the mitochondria. So, before that, the opening of a large conductance channel in the inner mitochondrial membrane, known as the mitochondrial permeability transition port (PTP), has been shown to be a primary mediator of apoptosis. So now let's look about each of these four models. So the first one is PTP induced mitochondrial swelling model, or also known as the VDAC opening model. So VDAC opening is triggered upon direct interaction of apoptosis inducing agent and PTP. So solutes and water enter, leading to a rapid loss in mitochondrial membrane potential, and also leads to mitochondrial matrix swelling and rupturing of the outer mitochondrial membrane. So subsequently, apoptogenic factors like cytochrome C, AIF, SMAC diablo, like we just saw in the previous slide, all these leak out into the cytosol. So the next model is PTP non-swelling model. So transient opening of PTP induces mitochondrial depolarization through a proton dissipation pathway in the absence of mitochondrial matrix swelling. So, this signals a conformational change in the cytosolic bags, leading to its translocation and formation of pores in the outer mitochondrial membrane. So, thus, like through these pores, the proteins inside are released into the cytoplasm. So the next model is formation of conducting channels model. So pro-apoptotic BCL superfamily members insert into the outer mitochondrial membrane where they oligomerize with each other, like the BACs or BID proteins, or they oligomerize with VDAC or other proteins to form conducting channels. So these channels are large enough for the release of some apoptogenic factors into the cytosol. So the last model is VDAC closure model. So closure of VDAC leads to the failure of the mitochondria to maintain ATP ADP exchange with the cytosol and a decrease in FOF1 ATP synthetase activity. So this induces hyperpolarization of the mitochondria, where mitochondrial swelling, rupture of the outer mitochondrial membrane, and release of apoptogenic factors are detected later. So in all of these mechanisms, the mitochondrial membrane potential decreases. And then another characteristic of apoptotic cells is annexin 5 binding. So annexin 5 is a cellular protein in the annexin group. So in flow cytometry, annexin 5 is commonly used to detect apoptotic cells by its ability to bind to phosphatidylserine, which is a marker of apoptosis like we saw, when it is in the outer leaflet of the plasma membrane. So it is usually the annexin 5 proteins are usually tagged with fluorescent dyes, and when bound to the phosphatidylserine of the apoptotic cells, they give fluorescent signals, helping us detect apoptotic cells experimentally. So we have just seen some of the characteristic features of apoptotic cells. And now coming back to our question, which one of the following is incorrect about a typical apoptotic cell? So option A, phosphatidylserine is presented on the outer cell surface. So this is true because we just saw that the lipids in the cell membrane are rearranged and phosphatidylserine is presented on the outer surface of the cell. So this is not the answer. And then option B, cytochrome C is released from the mitochondria. This is also true. We just saw how it is released and what it does to activate the apoptotic pathways. And then option C, mitochondrial membrane potential does not change. So this is incorrect because we just saw that the mitochondrial membrane potential decreases in apoptotic cells. So the answer to our question would be option C. Mitochondrial membrane potential does not change. So this is the incorrect option about a typical apoptotic cell. So yeah, the next option is annexin 5 binds to the cell surface. So this is true as well. Like we just saw in the previous slide, annexin 5 binds to the phosphatidylserine of the outer membrane of the apoptotic cell. So we have our answer. Thank you.